Hey guys, welcome back to the channel. Hope you guys are doing good. In this video, we will discuss the pathology of cell injury and try to understand it in simpler terms. So without wasting any more time, let's get started. Let's take an insight of cell injury. So what is cell injury? It can be understood as when a cell is exposed to stress or various kind of stressors like hypoxia being the most common stress, lipid peroxidation of membranes, DNA damage, oxidative modification of proteins, free radicals, etc. They cause cell injury. Now, the cell tries to adapt to the change. And if it fails to adapt, it leads to reversible cell injury. If the injury continues, it leads to irreversible cell injury, which further continues to cell death. So these are the events which take place during cell injury, and we will discuss each of them separately. Before that, let's discuss some important points. The most common cause of cell injury is hypoxia, that is low oxygen. And the most common cause of hypoxia is ischemia. Time for a little Q&A. So according to you, which cell is most sensitive to hypoxia? If you answered neuron as the answer, then you are absolutely correct. Now the opposite question. Which cell is most resistant to hypoxia? The correct answer to this question is the fibroblast. The function of fibroblast is formation of connective tissues. Now let's see the classification of cell injury. Cell injury can be classified as reversible and irreversible cell injury. Starting with the reversible cell injury. The pathogenesis of reversible cell injury starts with a stressor like hypoxia. The first cell organelle to be affected by hypoxia is mitochondria. So, hypoxia leads to mitochondrial dysfunction. One of the main functions of the mitochondria is to produce ATP. So, when mitochondrial dysfunction occurs, there is decrease in the ATP production. Now, the stopping of ATP production leads to three pathological processes: failure of the sodium-potassium pump, anaerobic glycolysis, and decreased protein synthesis. Let's discuss these three processes separately. Starting with the first process, that is the failure of sodium-potassium pump. As you already know that in a normal cell, 3 sodium goes out for 2 potassium into the cell. What happens in this process is there is reversal of this gradient. So, 3 sodium comes in and 2 potassium goes out of the cell. This leads to influx of water inside the cell and the cell starts to swell. This process is called as hydropic change, and it is the first morphological change in the cell injury. This hydropic change is responsible for cellular swelling, endoplasmic reticulum swelling, and formation of myelin figures. You may wonder what myelin figures are. Myelin figures are also known as concentric lamellation. Let's see why they are called as concentric lamellation. So this is the electron microscopic view of myelin figure. And as you can see, it is arranged concentrically. You may also have a doubt about the composition of the myelin figures. The answer is very simple. It is cell membrane, so mainly they are composed of phospholipids and calcium. This covers the first pathological process. Next is anaerobic glycolysis. You already know that the byproduct of glycolysis is lactic acid. This lactic acid decreases the pH, making it acidic. This acidic pH leads to nuclear chromatin clumping. Third pathological process is decrease in protein synthesis. Do you know what is depicted here in this image? Yes, it is the rough endoplasmic reticulum because it has ribosomes attached to it. What happens in this process is ribosomes get detached, and ribosomes are sites for production of proteins. So the protein production is decreased. To adapt, our body responds with a compensatory change, which is even more detrimental, and that is fatty change. All of these processes were in reversible cell injury. Now, let's have a look at the scenario when there is irreversible cell injury. So, how to know if it is reversible or irreversible cell injury? To answer this question, we need to discuss two classical moments of irreversible cell injury. They include severe membrane damage and severe mitochondrial damage. A bit of normal physiology here. As you already know, most of the calcium is outside the cell. What happens here is the cell membrane is damaged. So it leads to influx of calcium into the cell. The problem with calcium entering the cell is that it is a cofactor for activation of enzymes, and it does the same thing. It activates 3 enzymes: phospholipase, protease, and nuclease. You can guess by the names of these enzymes that phospholipase breaks down phospholipids, protease breaks down proteins, and the nuclease breaks down the nucleus of the cell. We will discuss about how this nuclease breaks down the nucleus and what are the changes associated with it. But before that, let's discuss the second classical moment, that is severe mitochondrial damage. So we discussed here that the membrane was damaged and calcium enters the cell. This calcium not only activates the enzymes but also sits on the mitochondria, forming amorphous flocculent densities. Let's see how the nuclease breaks down the nucleus and the changes associated with it. This is a normal cell. Let's assume the blue part to be the nucleus, and the pink part is the cytoplasm. First process is known as pycnosis, in which the nuclear chromatin condenses. Second is karyorexis. Karyo means nucleus, and rexis means fragmentation. So, in this process there is nuclear fragmentation. And lastly, the third process is karyolysis, in which there is complete dissolution of the nucleus. So these processes completes irreversible cell injury. So that is it for this video, guys. We will continue this in next part of the video. Don't forget to subscribe the channel and follow us on Instagram. Links in the description.